Hi, welcome to Detours, Understanding Acquired Brain Injury, and this is Unit 2, Chapter 1. We're talking about genes and chromosomes, and so this unit focuses more on actual production of proteins. So we discussed cells and organelles and a little bit about chromosomes. For this unit, we will continue to use the cooking analogy because when it comes to genetics, it really works kind of well. And genetics and cooking go well together because chemically they're not that different. So let's go back to baking chocolate chip cookies again. We all like those anyway, right? Back to the old family cookbook idea. As previously mentioned, the cookbook itself, the physical cookbook, is like a chromosome. Sometimes necessary to repeat these concepts, especially the concept as complicated as genetics. Many people haven't had experience with genetics, and it is a in-depth topic, and things change frequently. A typical family cookbook, as you know, it contains all the different recipes that you have. But right now, specifically, if we want to make chocolate chip cookies, if that's what you feel like, it's like you would have those favored recipes marked. And that in genetics, we refer to that as epigenetics, a way to communicate which recipes are used frequently or which ones are switched on or switched off. You can easily find the right recipe or the right gene because of epigenetic factors. Specifically, a transcription factor binds to the proper gene. Transcription factor telling you, hey, this is the gene we want to produce. Acts like that paper clip that you use to indicate your recipe. It tells you which gene or which segment of the gene, hey, this is start point for that gene, the transcription factor. Okay, the recipe has instructions, and this is important to understand. The recipe has instructions not only for how to make the cookies themselves, but it also contains instructions for like how to set the oven, how long to let things sit, what measuring spoons you should use, how many cups. It's like, should you let something sit out, grease the tray, all that stuff. And the same is true for genes. It tells certain RNA molecules, we'll get into that, where to cut and how many repeats, whether other genes need to be switched on, all that stuff. So a gene is a complete set of instructions. It's not just 2 letters or 4 or something like that. A gene is the entire thing. Most people usually think of just 2 nucleotides or 4. No, it's not. This is what a gene is. It's a complete recipe. I know we mentioned this in Unit 1, but it's important to stress that. So you don't look at 2 letters and go, oh, it's these complete set of instructions. And there are many, many more genes which regulate the function of other genes. And we often used to call these junk DNA back in the day. We found these are actually control and regulatory genes, which help in the production of, or which tell genes to bind and things like that. So most of the genes are not that. The idea of junk DNA is wrong. That is an old mistake. Most of those genes regulate the control and production from other genes. So just so you know. There's a saying in biology, basically, that everything in biology has exceptions. Basically, there's always an asterisk behind everything. And this, too, has exceptions, or every asterisk has an asterisk, and every asterisk has an asterisk. So just, You know that a gene, as I said, is a specific set of instructions, which includes instructions for coding for proteins. And these instructions don't always code specifically, but they tell the cell also how to put a protein together. And it will also tell the cell where to cut and assemble. In our analogy, as we said, the chocolate chip cookie recipe doesn't just tell you what ingredients you need. It tells you, what to do with them. And it also may tell you, what to do with another part of the recipe. Maybe you are using the cookies, the chocolate chip cookies, to make a gingerbread house for the roof or something like that. And so it may tell you other components. So it's important to understand how interactive genes really are, just to keep that one in mind, okay? There are also, these genes also for complete genes that code for actions or processes for what the cell is to do with the completed protein. So there are genes that give other sets of instructions for operation of cell systems too. All right, so yeah. Just again, to keep all of these kind of things running. So DNA as the building block of life, or the blueprint, you've heard that is accurate, but you just need to keep that just in your mind. Remember, DNA is really RNA, all of that is the story of proteins. Everything is built in your body from proteins, ultimately. Yes, there are bones and things, but they too depend on the presence of proteins. So what is a protein? A protein is a long chain of what are called amino acids, which bind together to form peptides. And the peptides, which are long chains, in turn then form these proteins, which are used to create enzymes and organelles. So an enzyme then is a protein that speeds up chemical reactions that, although they may take place, they just would be very, very slow. So an enzyme is critical because it speeds up the process of a chemical reaction without being used up in the process. So an enzyme will put things together or take them apart to cause chemical reactions to happen. So enzymes are critical for the functioning of a cell. Many enzymes include what are called coenzymes, and you may have heard like coenzyme Q10 or whatever. A coenzyme are typically contain a metal or metalloid from the periodic table, and they're essential to assist that enzyme in functioning. So a coenzyme will help make that enzyme able to break things down or put them together. Enzymes have a chemical range, a pH, for example, or a temperature at which they operate optimally, and other proteins help regulate those temperatures and chemical ranges in which they operate. Just so you understand, and those enzymes are necessary to allow these things to allow enzymes to function. All right, so back to amino acids. So they're organic acids. An acid is anything that produces or releases hydrogen. That is what makes an acid. We say pH, potential hydrogen. So it's and it leans towards the acidic side, which is more hydrogen. Amino acids, they're organic too, which you're looking for carbon, the presence of carbon in these. And there are different kinds of amino acids within that carbon group. They have two functional acid groups, two pieces. One is the amine acid group, which is built from carbon and nitrogen. Nitrogen is present in proteins. So if you see chemically a nitrogen group, you're dealing with a protein structure. Remember how I said amino acids are the building blocks for proteins. So if you see a nitrogen, an atom of nitrogen, you're dealing with, and you see carbon like this, you're dealing with an amino acid. Okay, this is an amino acid. Okay, and the stem of this amino acid is formed from this carboxyl group. This is part of this amino acid. Okay, and then there's what we call an R group, and you change these stems, this part of this group, and you could put together different amino acids using these changing R groups. It's just the name of this stem. So amino acids link together in long chains to form a protein. And then, because of the way they carry an electrical charge, they will fold into all kinds of many different structures. There are hundreds of amino acids, but only 22 of them are found in cells. Only 22 of these guys are found in cells of living things. But you can block and chain them all together and make all different kinds. And they can perform, and you can plug proteins together to form even more complicated structures. And when you connect a chain of amino acids together, you form an intermediate structure before it folds called a peptide. So when you chain them together, you get a peptide. When they fold, either by themselves or with the assistance of enzymes, then you get a protein. All right, so let's talk about how this process unfolds, as it were. All right, RNA is what's called a polymeric acid that performs almost all the major active functions of cells, regardless of where you find them, whether it's in a bacteria or whether it's in a person. RNA is typically, but not always, a single-stranded structure. Looks like this. It is a close cousin of DNA. It probably emerged first. The working RNA is referred to as non-coding RNA. Coding RNA transfers information, is like kind of managerial, if you will. It conveys information. Non-coding RNA does something. It does a job. It's the working RNA versus the boss, okay? The management, okay? That's an easy way to think of it. There's more to it than that, but still. So non-coding RNA, there are a lot more of non-coding RNA types than there are of the coding RNA. The other way to think of coding RNA is like programming, giving instructions, a computer code, which is where the idea comes from. Non-coding RNA performs functions. Programming versus performing the computations. All right. There is DNA in the nucleus of cells, of course, and that is double-stranded. But you find the DNA in two major places. You find the DNA in the nucleus of the cell. Remember, this is DNA. And it's similar to RNA in a lot of ways, to coding RNA. It is double-stranded. It's that infamous helix that they talk about, although some recent studies suggest there's a lot more to it. It's not just that famous double helix. It may form other shapes as well. And there is DNA, which forms a ring that is in the mitochondria, mitochondrial DNA, which we did talk about. They call the DNA in the nucleus nuclear DNA because it is in the nucleus, and DNA that is in the mitochondria is mitochondrial DNA. Okay, we will take a brief break here, and we will pick up again in Chapter 2, Unit 2. Remember, please click like and subscribe. Don't forget the bell icon. And leave any comments below. We'll see you again soon.